Hi, my name is Melina Hale. I'm a professor at the University of Chicago. My lab works on neurobiology, biomechanics, and evolution. In a previous video, I gave an introduction to evolution. And now I want to give you an example of how we study evolution in our lab. And one of the things we look at is the evolution of neural circuits. So I'll talk about one of the systems we study, which is the startle response, its neural circuits, and how we think they're evolving over time. So the example on the screen here is, you might be able to tell it's a fish. It's actually a larval zebrafish. It's about 5 days old and 3 millimeters long. And the probe you see coming in from the left side of the screen is a little injector, and it has dye in it. And one of the ways we get fish to startle, to study this behavior, is through puffing a little bit of dye at their body and then watching their response. So you'll see its response. It's analogous to startle in humans, which might be if you're at a scary movie and something surprising happens, we jump. This is the fish equivalent of that. It looks a bit different. Let me show you the video, and we can talk about it a little bit. So that's the startle. So you might have been able to see the dye hit the side of the head of the animal, and then you see this big turn to one side, and the animal swims away. This is the behavior and circuit we'll mainly be talking about today. So let's back up for just a minute, though, and talk about how we study the brain. A lot of different researchers are working on brain function and structure, and have been for many, many years. And there's a lot of beautiful work going on. Some of it tries to identify how regions of the brain are functioning. So here we see half of the brain only in a translucent skull. And in red is one little part of the brain. That's called Broca's area, and you'll see it when it comes back around right there. This is actually part of the brain that's really active when we are speaking. So as I'm talking to you, that part of my brain is really, really active. And people go and study all these different regions of the brain to understand when they're active in relationship to behavior and how they interact. So in addition to understanding the cells that are in these locations, like in Broca's area, researchers are also trying to understand how these cells connect. So the cells in the nervous system are not just little balls like you might see in some typical pictures of cells. They have long processes on them, and those processes connect between cells, from cell to cell, and among regions of the brain. So what we're looking at on this image is lots of different processes from regions of the brain, from particular neurons that are traveling to other parts of the brain to integrate information, to transmit information, and ultimately to generate some sort of function, whether it's a behavior or storing memories, various things like that. Another way we've been studying the brain, in addition to doing experiments that try to look at connections and regions, is through wonderful neurology work that's examined how injuries are associated with changes in brain function and behavior. And that work started a very, very long time ago. This is pages from the Edwin Smith papyrus, which is from about 1500 BC. And is the earliest documented incident of people actually relating an injury to the nervous system or to the body to a behavior. So seeing that there's an injury to a part of the spinal cord or brain and how that affects the movements of an animal or the behavior of a human in particular in this. In my lab, we're interested in understanding how particular neurons, nerve cells, connect to one another, and how those cells and the simple circuits they're a part of generate movements, the startle movement in particular that I showed you earlier. So to understand circuits and how they function, a lot of us use simple model systems for that work. They're much simpler, less complicated than the human brain or large brains, but yet they let us get at basic principles of how the nervous system functions. One of the fantastic models that's been used for neural circuit work is the nematode, C. elegans. The nematode is pictured right down here. It's a tiny little worm, really, really small. It has only a small number of neurons in its nervous system, a few over 200. And folks working on C. elegans have been able to map how those neurons connect to one another. So what you're seeing in these little blips to the left of the nematode are different neurons and their pathways, the connections from one to the other. Now, there are a number of other simple model systems that have been used productively in neuroscience. The sea hare on the left, lobsters, lampreys, and bees are just a few examples of those. And they've provided an understanding of foundational principles of connectivity and function, really important things that have influenced a lot of work in neuroscience. For example, the work in learning and memory in the sea hare on the left. When you're interested in evolution, though, as we are, we still have some issues here. So we can have all of these simple models spread across the phylogeny, but because there's so few of them and they're so far apart, trying to reconstruct the evolutionary history of a neural circuit is really difficult. And we're just at the beginning stages of being able to do that in organisms. The startle behavior, for several reasons that I'll talk about in a few minutes, is one of the best systems for understanding how neural circuits evolve. Part of the reason that it's so useful is that because the startle behavior occurs in so many animals across the phylogeny, it's a key predator avoidance behavior. So you might imagine evolutionarily, it's going to be very important and highly conserved. What we're looking at here is a pike, northern pike, on the image on the right, and what you're seeing on the left is a still from a video that I'll show you in a second. And just to orient you to that video, we're actually looking at the belly of the fish, the ventral view, and that gray shadow above it is my hand coming over, and I'm going to startle this animal. So if we look at the movie of that behavior, you'll see how the pike startles. So let's get going. There, I touched the fish on the head, and you could see it did that really fast movement away, like the larval zebrafish in the first image that I showed you. Okay? Now, this type of startle, this bend to one side and swim away, is characteristic of many, many species of fish. If we look at it in just a set of silhouettes, we can really break down what that behavior is. Generally, these fish start in a straight position. So as they're sitting in the water, not expecting a predator to come along, they're just hanging out and their body is straight. When they sense a predator coming from one side, as shown in image 3, they do this massive turn and bend. They orient their head away from the predator, and then they use a number of swimming strokes to get out of there as fast as they can. So this is a typical response, as I said, of a number of fish, including zebrafish and pike and many others. Now, what's driving that behavior? Let me tell you a little bit about the neural circuit that's important for that response. The circuit is highly conserved, so we see some of the same neurons in pike and in zebrafish and all these other species, even though they're hundreds of millions of years separated in evolutionary time in terms of when they diverged. So this is, you might not be able to tell right away, but this is the brain of a zebrafish with certain neurons labeled. So just to orient you, here are the eyes and the ears of the brain, the otic capsules. And all those white blips are different nerve cells that are in the brain and are sending those processes, what are called axons, down into the spinal cord of the animal. So they're sending signals down to the body to get the fish to do something. This is a larval zebrafish, so the head is only a millimeter or so across. Now, these here are really important cells for the startle response. They're called the Mauthner neurons, and there are only 2 of them. They're giant neurons that are in all of these fish that do this type of startle that I've shown you. And they're located in the hindbrain of the animal. Up here are a different set of neurons we won't be talking about, but they're neurons in the midbrain that control that rhythmic swimming that you might have seen at the end of the videos that I showed you. Okay, so we're going to focus on the Mauthner cell and the Mauthner cell circuit. So this is a simplified Mauthner cell circuit. So the Mauthner cell and the neurons that it connects to. I just wanted to show you a little bit about how it works in the organism. So if we have a stimulus in this circuit coming from the right, So here's the circuit. The stimulus is coming from the right. The Mauthner cell on the right, which is this big dark blue cell, is going to fire. When it fires, it's actually generating what's called an action potential, a signal that's transmitted all the way down its process, down its axon. In this case, it goes from being in the hindbrain, which is that top group of cells on this image, down into the spinal cord. In the spinal cord, it'll signal other interneurons and motor neurons to signal muscle to cause the body bending that I showed you in those videos. So if I'm the fish, and this is me, a stimulus coming from this side of my head would send this Mauthner cell, cause the Mauthner cell to fire. It would send a signal down my spinal cord, and I would bend in that direction, forming that C-shaped bend that you saw in those videos. Now, one of the really interesting things about the Mauthner cell is that there's this special part of the circuit that's really not diagrammed in detail here. It's called the axon cap. And the axon cap has all sorts of interesting things going on in it. It has cells coming in that excite the Mauthner cell and get it to fire more readily. It also has neuron processes coming in that inhibit the Mauthner cell and prevent it from firing. So there's a lot going on in this region. Because the Mauthner cell is so big and obvious and so conserved across a lot of taxa, we can use the cell as an anchor to understand those circuit components that are in the axon cap structure. So let me show you a Mauthner cell firing, and then we'll talk a little bit more about it. So this is a Mauthner cell that I've injected with what's called a calcium-sensitive dye. So when the Mauthner cell fires, calcium floods into the cell, and the dye actually reacts with the calcium and becomes brighter when the cell is active. Now we've color-coded brightness with this heat map so that when the cell fires, you'll see more reds and oranges. So here's the Mauthner cell, and you can imagine I'm tapping the fish on the head from this side, and you can see what happens. So that's a Mauthner cell activation. That's the activity that's initiating that big bend that we showed you. Let me show you one more time. I touch the fish, and it fires really, really quickly. Something I didn't point out before is that these various videos I've shown you, and even this image, is slowed down quite substantially. A typical Mauthner cell response, the behavior would occur within tens of milliseconds, so in less than the blink of an eye. So studying the Mauthner cell overall, and this is not my lab specifically, but in this broad group of researchers that have looked at the Mauthner cell over 100+ years, we've learned a lot about neuroscience. The simple system has been really, really valuable. We've looked and been able to understand more about the subcellular structure of vertebrate neurons, how neurons communicate to each other through different types of synapses. We've learned about this transmission of electrical signals between neurons as well. In addition, the Mauthner circuit has been used to study learning and to look at the relationship between neural activity and behavior. So it's given us access to circuits in a way that we just can't get in the human brain, for example. All right, so let's talk about a little bit about evolution of this circuit now. So I introduced you to the Mauthner cell and told you that it's found in lots of different animals. And I also mentioned the structure called the axon cap, which has excitatory inputs and inhibitory inputs and gives us a little window into how neural circuits might work. So this is a phylogeny that I showed on a previous video that I did on an introduction to evolution. It's, again, of just vertebrate animals, and only a very, very few of them, where we see on the left the jawless fishes like lampreys. We move up the tree through the chondrichthyans, into fishes, and then tetrapods as well. Now, for the Mauthner cell, we see its presence in many of these different groups. Lampreys have the Mauthner cell, chondrichthyans, and a few species we see Mauthner cells. Many, many fishes, the vast majority of the 30,000 species of fishes that are known have Mauthner cells. And even in amphibians, in both tadpoles and in some frogs, as well as in salamanders, we also see Mauthner cells. When we move up into the tree, though, we have quite a mystery to solve. We do not know what happens to these giant, really important neurons once we move up into the tetrapod animals that have become more terrestrial and have changed their startle behaviors. It's one of these big questions that I'm so curious about in my labs trying to investigate. So let's look at that cap and see where we've gotten in terms of understanding how the Mauthner cells and their circuits have evolved. So remember where those yellow stars were on that very simplified diagram of the Mauthner circuit. This is what we're looking at here on the left, that cap structure. And as I mentioned, they're excitatory neurons that are connecting to the Mauthner cells. Those are shown as these cells that are this process that's spiraling around the base of the cell on the far left. Then we see this sort of egg-shaped capsule around that process of the cell. And those little black nubbins coming in are the inhibitory interneurons that are shutting down the Mauthner cells. So we have excitatory and inhibitory components. On the right, you can see what that actually looks like in a section of a brain of one of these fishes. So this is what we call the composite axon cap. It has all of these different components to it. This is the cap that you would see in fish that if you go to an aquarium and are looking at a lot of fish swimming around in a tank or at a pet store, most of them have Mauthner cells that look like this. Now, I showed you this image of the startle at the top panel here. This is shown in a trout. And the composite cap and Mauthner cell generate this type of startle. But startles in fish are actually a bit more diverse than that. And in particular, if we look down in the lampreys, which diverged off of the main lineages of vertebrates at the base of the tree, we see a very different type of startle. It's a retraction response shown down below here, where the animal actually, when stimulated, kind of contracts or accordions its body. In lampreys, the Mauthner cells are there, but they don't have this axon cap structure. So without these inputs, they generate a very, very different behavioral output. So here are the two primary basic types of caps that have been known for a while. This composite cap of typical teleost fishes, typical bony fishes, and this no cap condition that we see in these retracting animals like lampreys. In my lab, one of my students, Hilary Bierman, and a colleague, Steve Zottely, worked with me to try to get a better sense of how this cap structure varies across species and whether there is actually variation between those two basic morphs. When we looked at lots and lots of different species, we actually found that there are 4 types of caps. In addition to those first 2 that I talked about, we have what's called a simple cap that's shown on the top, where we have those winding excitatory fibers at the base of the Mauthner cell. And then we have what we're calling a simple dense cap, where we have those same winding fibers, but they're much, much more populous, much denser in the axon cap region. Note in both of these cases, in these images and the diagrams shown here, we don't have that nice glial, the nice cap around the axon, where we have those inhibitory interneurons coming in. So they differ quite a bit from the composite cap. So let's map the Mauthner cells and the Mauthner cell axon cap onto the phylogenetic tree of vertebrates. And this is a bit more of a complicated tree, detailed tree than what I've shown you previously. At the base, shown in yellow, are 2 clades that actually even aren't vertebrates. We see the Amphioxus there and hagfishes. Amphioxus and hagfish actually don't even have Mauthner cells. We think the Mauthner cells arose before the lampreys, which are shown in white, branched from the rest of the vertebrates. Now, as I said previously, the lampreys do this retraction response, and they don't have axon caps at all. As we move up the tree, we see several lineages that are shown in purple that have a simple cap structure that I showed you. Those include things like sharks, and as we move up, amphibians and lungfish, as well as some of the basal bony fishes called bichirs and rope fishes. Moving up the tree from there, we see the evolution of the simple dense cap. And that's in these species that we don't actually have a lot of extant today. There are some species, but not a ton of sturgeons and gar, and there's really only one extant species of bowfin that was a very diverse group many, many hundreds of millions of years ago, they all have the simple dense cap. Interestingly, where we see a transition is into this composite complex cap structure is right at the base of the teleosts. Now the teleosts are the modern bony fishes. It's this amazingly diverse and speciose group. There are many, many species. It's the dominant group of fishes in the world today, the extant fishes. Nearly all of them have this composite cap. It seems to be characteristic of that group of organisms. Now, evolution isn't quite that simple, and we do see some differences from this nice pattern that we've looked at up the tree. In particular, if you go back over into the purple area where the sharks are, a branch in that tree called the ratfish or the chimeras have a simple cap. And in the teleosts, you can see that one white branch, and that's the eels. And so a few places through the tree, we will see variation. Now, if we look at how the cap structure and this variation relate to behavior, if we map behavior onto this tree, we can pull out a couple key points. The first is that when we have the absence of this axon cap, when you just have the Mauthner cells with none of this excitatory and inhibitory inputs coming to them, we tend to see a retraction response, which is that pulling back of the head. We see that in lampreys and also in the eels, which lack the axon cap structure. All of these other animals perform more of a C-start type of response, although it varies in its strength and its how high performance, how fast it is. But one thing that's noticeable is that when we get to this composite cap at the base of teleost, we see a really high performance response. There's some innovation of how that cap works that allows the performance to be very, very high, very strong response. This middle area of the tree, we're still figuring out. Fish with the simple and simple dense caps have more diverse startles, the same basic pattern, but there's a diversity in performance and how it's generated by muscle that we're still trying to understand. And the motor patterns are quite complex. Now, if we get up into the amphibians, as you could imagine, I mentioned earlier that not only do the tadpoles have Mauthner cells, but also frogs. You probably can predict what a frog's startle response will be. It'll be a bilateral hop. One of the things we're trying to figure out now is how that relates to the Mauthner cells and this circuit. Well, what about the startles of humans and other mammals? This is an example of an armadillo startle, where the animal jumps up on its legs into the air when it's startled. Are these types of behaviors in our startles also generated by Mauthner cells and this Mauthner cell circuit? We actually don't know. There's similarities in the startle behavior, as you might imagine. They're really fast, really high performance, but they're different, and we haven't been able to associate the neural circuit of the startle in mammals with that of the startle of fishes. It's one of the things we really want to know in my lab and are beginning to take steps to figure out. So let's conclude. First, I want to make the point that there are a lot of ways of understanding how the brain works. Lots of labs doing beautiful imaging work, studying healthy human brains, and also studying injured human brains where we can associate damage to a particular region with changes in behavior or function. In addition, we can use more tractable brains or more tractable nervous systems in other species like C. elegans and the sea hare to understand how circuits are put together. Looking at those simple circuits, we can use them to understand basic principles that also may apply in tetrapods and in other vertebrates. Now, in addition to using models, comparing across models or other species gives us a different sort of power of looking at how things are similar or how evolution may have come up with different solutions to generating certain functions in the body and of the brain. So we use comparisons and evolutionary approaches to relate structure and function more broadly. Now, on the Mauthner cell system in particular, I hope I've shown you that the Mauthner cell is an example of a simple neural circuit that we can use to understand not only basic principles of the nervous system, but also nervous system evolution. In particular, because we can follow this cell and use it to anchor the circuit through brains of a wide variety of animals, we're able to then track the circuit over a large swath of evolutionary time from the divergence of lampreys on up. And that divergence occurred about 500 million years ago. So we're talking about a really long time frame here. So as I end, I just want to acknowledge some of the people who are important to this work. In particular, some of my students. These are all students who were in the lab over the past decade or so. Hilary Bierman, who's a graduate student, and Rachel Fremont and Julie Schriefer, who are both undergraduates at the University of Chicago. In addition, my collaborators at the university and at other institutions include Steve Zottele, Mark Westneat, John Long, and Vicki Prince, and they've all been important to the ideas as well as the data collection and analysis of this work. In particular, though, I'd also like to thank Glenn Northcutt. The images of axon caps that I showed you, some of them were made in my lab, others were actually photos of collections that he's taken over a number of years. And they've been so important for our work. So I wanted to thank Glenn, and also to make the point that collections, biological collections, are just really important, not only for what we've learned up till now, but for the future of discovery in science. And of course, I want to thank our funders. The National Science Foundation and grants that we've had for work in the lab have been really important to our research and our ideas. So thank you.